Yeah, so we are around 20 at the moment. We are going to begin slowly and then to wait the last few persons. So thank you for everyone to be here and thanks for your time. Good afternoon with us. Cyril Lefranc from the Society Planck-Tovey here at the University of Science of Montpellier in south of France at the Laboratory of Pathogen and Host Interaction with Madame Catherine Gonzalez.
Bonjour à tous.
She's very pleased to welcome us at the Zebrafish facility here, and we are very happy to be here with her. So we are here to show you first of all, and first time, our feed dispenser, the dry feed dispenser, the fish gun for the first time, then the liquid feed dispenser, the fish gun also, and then a new device, the Spin Touch, to be able to make 7 tests of the physical and chemical parameters of your water, like the phosphate, the nitrate, and many others. And so I begin with and then and then we will show you with Antoine Barbeau from the company Viewpoint. Its device, the Zebrox, to be able to follow the behavior of the zebrafish all around the different life stage of the zebrafish in the last part. So thank you again to everyone to be here to listen us. So first of all, I show you the fish gun for the dry feed dispenser with a tube where we have put some dry feed, the Gemma Micro 500. So it works with the vibration and with 3 modes. So the manual mode. So as long as we feed the fish gun in front of the tank with the fork in the front, that will make a vibration like this. So as long as you make working the fish gun going to deliver dry feed, then you have a fixed mode. Then you fix the number of fish you want to feed. For example, 5, and it's going to deliver 5 portions, and then it stops to work. And at the end, you have a RFID mode with an RFID antenna at the front of the fish gun. It is going to read some RFID tags like this, where we have encoded the number of fish we have in each tank, and the fish gun is going to read the tag and to deliver the right amount of feed for each fish in each tank. So just like this, to show you. Just like that. I have no effort to do. This is the fish gun is working, and putting the feed inside the tank by the hole on the lid, like that, without any effort for those technicians. So that allow you to avoid any pain on musculoskeletal pain. So just like that, it's very reproducible, very accurate, and you can design the portions for fish with a menu, the menu time per unit. So you design the time of vibration that the fish gun is going to deliver to feed the fish. For example, for the Gemma 500, with 100 milliseconds of vibrations, you are going to deliver 9 milligrams of dry feed for each fish. So you can, like that, design precisely the portion you want to deliver to each fish. And if you make 2 or 3 runs to feed your fish per day, you can just divide this time of vibrations to precisely calibrate the size of feed you are going to deliver per run. So then we have different tubes, tubes with a hole of 2.4 millimeters, and a tube with 2.0 millimeters at the bottom. The 2.4 millimeters is at the right size to be able to deliver dry feeds between 300 and 500 micrometers, and the other tube with the 2.0 millimeters of size hole is ready to use to deliver a dry feed between 75 micrometers to 200 micrometers. So like that, you are able to feed all the developmental stages of your fish. And this even you have different persons that they are working on your facility on feeding your fish day after day or during the weekend, or if you have different zootechnicians, different teams. Then this is easy to program RFID tags many times with, for example, this RFID reader on encoder that you can program and read, write, and rewrite many times, change the number of fish you have on the tag as you want. And you can also do this with some apps like NFT tools, or we are also working on a new app from Planturvy to help you to program your RFID tags very easily and avoiding many steps to keep your time for time saving. So is there any question about this part? First of all, I'm going to see. Can you use the feeding system also for a different system? We have different tanks. Yeah, I understand. Yes, for sure. I avoid to precise this. You have seven position of the tube you can put on the fish gun like that, and just like that you can put the tube at a distance, different distance from the tank, and depending on the brand of your tank you are using, you can with that adapt the fish gun to the many brands of tanks you have in your facility: Aquaneering, Techniplast, Aquatic Habitat, and many others. Iwaki also. I'm working on that. So yeah, this is adaptable to many different brands of tanks. Just looking for some other questions. Yeah, we can send you. It is possible to test the feeding system. We are able to send you a demonstrator to let you use it for 15 days, and let you try the time to let you the time to try it, and to purchase it if you like it. So now I am going to show you the light feed dispenser. Oh, just a minute. So it's working with the tank like this. We have put it inside some light feed like Rotifer, like Artemia, like Daphnia. We have just in front, just inside layer pump to mix the light feed inside the tank and to homogenate the light feed solution to be sure you have the same quantity of light feed everywhere in the tank. We switch on the tank, and it works also with a gun like this. We put the tube in the top, and then we have all the same menu, the same mode, manual mode. So as long as we make it work, the fish gun is going to deliver like this. We have a fixed mode for a precise number of fish, for example 10, 15, and then it's going to deliver 10 or 15 portions, but it's workable until from 1 until 99. And we have the verified mode, like for the package dispenser. It's going to read the tag and to deliver the right amount of feed for each chunk, like that. So this time this is not a time of vibration, but a time of pumping. And depending on the concentration we have of light feed we have inside the tank, we deliver a precise number of light spray to each fish. So for example, we have a program for 48 milliseconds of pumping, and so that deliver 1 milliliter per fish in this case. So like that with the right number of light spray inside the tank, we can adjust depending on our feed protocol the number of light feed we want to deliver to each fish. So yeah, just like that. And this works also yes with the RFID tag as with like with the vertical dispenser. Just put it down and get you wait for some questions. Okay. There is no question. Okay, so you have any question maybe later, don't worry, and feel free to contact us. So I'm going to show you the next device. So show you on this side. So here, screen touch. Like that. So this is the photometer screen touch. It's work with this kind of this. So with different parts with different reactives for all the tests. So just with one sample of water and 1 minute. Just when you use it. So you can like that do 7 tests of for your water at the same time. Let me show you. A sample of water of the system. Just filling the disc like that. And avoiding any bubble of air inside. Just slowly and filling the disc just maximum 10 minutes before the test. for the best accuracy. So you can just put the cover disc like that on the inside the photometer with the cover disc. Then we switch on. Yeah, just like that. Select the correct type of water, seawater or fresh water. In our case, this is fresh water. And the correct number of this with the FX104. And then let's go to start it. So it's going to work just 2 minutes. And at the end, we will have the can test certain results at the same time for the pH, for the ammonia, nitrate, nitrate, phosphate, alkalinity, and the pH, yeah. So yeah, and that's it. And this is very accurate, and the device is making some corrections between tests to avoid any error of testing. For example, for the ammonia, there is some check with the pH and the alkalinity to make some correction and to deliver the right result for the test. And when we do test separately, we don't do this kind of correction. So like this, we don't have to make some work. We can do another thing in the same time it is working, and we don't pass 30 years to make the current test, for example. This is very quick, very time saving, and we have nothing to do. Then this is we bring it with a disk of calibration like this one, and after that you can also export the data of the results of the test. It is to your computer, which is white arrow, so just like that. So just wait 2 seconds more, and we are going to start the results. And yeah, so that don't take too much place on the table. It's very fast. So yeah, for example, there we have some fuel ammonia 0.7 ppm, some fuel nitrate 0.1 ppm, some nitrate, some phosphate, because we have Zebrafish in the facility. Yeah, so we have a pH of 7.2, etc. So yeah, so it's very practical, very easy to use. So do you have any question about this device? Okay. So thank you for your attention. Now I let the voice to Antoine Barbeau from my colleague from Newport. Sorry, that's a few minutes for the question. Yeah, we can send you a price list for the different devices. We will send you this price just after the presentation. And if you have any questions, don't hesitate to contact us by email on the website, for example, with the contact platform, or just give us your question in the chat, and we will send you answer after the presentation. So yeah, I let you, Antoine Barbeau from Society Viewpoint, to show us your device for the behavior of the Zebrafish.
Thank you, Cyril, and thank you, everyone. I hope you are hearing me well. So if you don't, just let me know. So we'll just share my screen. So I hope you can all see it now. Okay, perfect. So I'm going to present you the company Viewpoint and more precisely the different behavioral tracking solution we have for the different zebrafish stages, from embryos to adults to larvae. I say standard and custom-made solution has most of our devices that we have in the market came from the lab's request directly, and we try to fit your needs. So that's why I'm going to present it that way. So just a brief information about the company for the one that doesn't know the company. So Viewpoint, it's a French-based company up north Lyon. we also have like an office in Montreal and another one in Shanghai to cover worldwide cover. We started in the 90s with the first video tracking system for rodents analysis, so mice and rats. And then in early 2001, we got a request from the US to develop a system for zebrafish monitoring in high throughput, so up to 96 larvae simultaneously. And it's how we started in that scene with our Zebrox system. And since that time, we keep improving our system to cover all the different stages and to add some different stimulation depending of how deep you want to go into your research studies, your protocols. So that's why we are always keen to know about what you want to achieve and how we can help you out. And as information, Viewpoint is also present in different markets now for the phenotyping of plants to optimize the growing of different plants according to the different world areas they are. And more recently, we also have a dedicated system for the water assessments based on the video tracking of aquatic microinvertebrates and their response being touched with different micropollutants that can be based and present in the different industries or waste water treatment plants and drinking water treatment plant as well. So that's why we diversified our market and product lately. So to start, I mean, our main system is called the Zebralap Software. As you can see, we represent the software through that screen and the heat map. So we deliver data output, just distance, locomotion on larvae, embryos, and adult zebrafish. And the software can be plugged and connected to different hardware. So where, as all our systems are based on video tracking, so we have different equipment depending what you want to do on which development stage. So we have the Zebrox for embryos and larvae. And then we have what we call the Zebra Cube for adult zebrafish. In a control environment has the Zebrox, as you can close the chamber. Being in a control environment, as you can control the light. You can also add some module to respect the circadian cycle and so on. And the Zebra Tower, which is a derivation of the Zebra Cube, but you see in an open standalone, meaning that the fish will be under the condition that you have in your room. So if it's not a point to control the lights, that's totally fine. So we have different approach and system depending on what you want to do. So the main things of the software is the fact that you can launch and assess the zebrafish behavior in live. So meaning that straight when the fish and the organisms are ready to be tracked, you just place on the hardware, Zebrabox or ZebraCube, down the camera, and we perform the analysis. In parallel, we will record the AVI file. If you want to replay the data, if the D-Day, some parameters you figure out that they were not well parametered, you can rework on the raw file that we provide, as well as the video file on your organism that were ready at the day, because they were at the correct stage. They got the concentration or the thing that you wanted to analyze at that day. So the thing that you can work in live up to almost 400 larvae simultaneously, as you can plug up to 4 Zebrabox to 1 Zebralap software. So you can work in live with 4 times 96 larvae for really high throughput for toxicology assessment, pharmacology, for example. So I'm going to present you after what can be plugged on the software. And just for information, we keep improving the software depending on your request to create something that can ease your daily routine using the software by creating animal lists. That way you have more outputs on the final data sheet with for example, the half of the plate was your wild type, and the other one was your maintenance or different concentration, to try to filter the data and help you that way. Then most of the labs use their own script and so on. So, but when we can help that way, we can easily improve it and from time to time to release some new version. So the key product is our Zebrox. So you have a video, camera video at the bottom of the box, and then with 2 mirrors system, you have enough focal distance between the camera, the 2 mirror here, and the macro plates that you place here. It can be 6, 12, up to 96, any standard macro place that you place. And then we record in live what's happening. On the Zebrox, as a standard module, you have underneath the macro plates, you have a white light system. It can be used either for light pulse, just to see how the fish react to those light stimulation, or just to have a control environment and to get rid of external bias in order to have only the behavioral pattern of the zebrafish based on what you want to test, excluding all external factors such as ambient noises in the room or light and dark. I mean, depending on the room you are using, but just to control the environment. Here on that screenshot, you can see how our stimulation module looks like. Indeed, on the Zebrabox, you can have many different options that you can add on the Zebrabox later on without any problem. For example, to control the lights, to perform some optogenetic light sequences, some temperature change, vibration units, and then all those stimulation can be easily set up on our software. So you see here that you have 3 different stimulation possibilities here. So the backlight being the white light device down the macro plates, the top light. So we place a top light stimulation with 2 arrays of LEDs at different wavelengths that are on top light label. So all the plates are under the same condition when you trigger those optogenetic lighting. And the third one is vibration. So we stick a speaker behind the plates, and then you just enter the different values depending of the protocol design you want to run. So you just enter the duration of the different sequences. For example, let's take the backlights. So it means that for 1 second, you will have, let's say for 10 seconds here, you will have no light, then 1 second a pulse, then no light, then pulse, and so on. And you can synchronize the locomotion outputs, which is the framework of the software to give distance and locomotion activity on the larvae along different stimulation. And that way you can see precisely what were the locomotion response on each of the cycle. So I know exactly what the larvae were doing when my light was off, what's happening when my light was on, and so on. So you can either trigger it that way, or just have the total distance travel or activity generated per organism throughout the experiments, and having those stimulation just to have a control environment. So you can use both. So it's called the cradle mode. So either you base your data outputs on the stimulation, or just to have a control environment, and you just want to have the data out of the along the experiment itself. So the Zebralap tracking, there are 2 main options. So the tracking, so we'll follow the gravity center of the larvae swimming around the well, and you can classify it by discriminating the different activity levels. So small activity, average activity, and high activity. So it's mostly used for larvae and adult fish as they are moving forward. So briefly, I will let you watch that video stream from our software to let you know how you can set up a protocol. So the idea behind that is to automatize the design protocol. So what you are seeing right now, it's something that usually you just need to create once the protocol is done and you want to use it all along with your research or your studies. You will just save it as a protocol, and when you will be working on a daily routine on that, you just open that protocol, you have all your areas designed, and then you just start the experiment and get the output. So as you can see, we always create 3 areas. So the top left of the macro plates, the top right, and the bottom right. And as soon as you have done that, then we create automatically the entire plate. So now we are on a 96 well plate based, but it works the same for 6 well plates, 24, and so on. It's always the same process. Then you just play with the detection threshold, which is the most important features, in fact, to see precisely the red dots only on the larvae, to detect precisely and not to take any artifact that could occur during the experiments on the wilds. For example, I don't know, some artifacts or some excrement that the fish can release throughout the experiment. And for stigmotaxis assessment, you can also create in an automated way some inner area to see if the larvae are rather staying by the wall when they feel they have kind of stress level, or rather crossing the wall and so on. So the data output will give you the time spent in any central areas, the time spent, the number of time, and so on. And once you save that protocol, you just open it, run here, experiment start, and then you can work in a daily routine with all the plates you will be working with. And then the second software module, part of Zebralab, is the quantization mode. So here, that one stands more for embryos assessment, or larvae that are kind of static, so they don't move forward. So it means that their initial starting point will stay the same. They will not move forward. So we cannot track them through the gravity center. So it won't be pertinent to use that even with larvae if they are not moving forward. So here we will be able to precisely to monitor those discrete movements by making a difference between one image and the following, based on the image acquisition process. So we record at 25 FPS. So we will make the difference between one image and the other. And when the larvae or the embryos, they move the tail, or they are in the stroke or seizure stress, we will be able to see those discrete movements. So here is the same. You see a mackerel plates. So that one is a circle one, so it was the same. You create the 3 areas, then we will create automatically the entire drawing area around each well. And here you can see that we are now working on embryos, so they are not moving forward, so the tracking won't give any pertinent information. So using the quantification mode, you will see afterwards. Here on the right side, you can define, so the unit is pixels, pixel based. So you will just enter the threshold, meaning that in that example, all, let's say, movement or activity beyond 2000 pixels during this timing will be considered as a burst. So I just here reduce the units, and now you will see on the chart in live that gives the first readout of the activity per well, and you see the different activity levels. So you see a color code, white being the low activity, green medium, and from time to time, depending on the threshold you use here, you can see some red color code, meaning that they were in burst. So you cannot see here as you could see on the tracking, but as soon as the embryos move, you see some red dots on the place where the embryos moved. And it's that way we can quantify a fine activity. And then we have different software options that we can install on the software based on what you want to analyze, such as the shoaling. That can be really interesting to see how the shoal, depending on different approach, you want to see how they behave together. So that mode will be to analyze the group itself. We won't give individual information on the locomotion. We will give an overview about how the shoal behave with each other. So the distance between one individual and the rest of the group, the nearest neighbor distance, the polarization, so toward which direction they are all swimming toward. And for your information, we are working on a similar approach to be able to track on a group-based shoal individual tracking information, because we need to change the way we code and the way we analyze it, because with standard acquisition tracking process, we cannot guarantee when they're crossing each other because of the depths of the tank. We can lose the path of each of the fish. So we need to see from this information from another perspective and to work on that. So we are doing it. So I hope it will be released soon, as we know that by talking to you on either in live during some congresses, I mean, back in time, that it's something it's important. So we are working on it. And for information, we can place up to 10 individuals within the same tank, even more to assess those shoaling approach. It can be for most of the time adults, as maybe it's with the age and the development that fish start to behave as a shoal. With larvae, it's also possible. Then I'm not a scientist, so I don't know exactly how at the larvae stage, how a shoal analysis can be pertinent, but at each stage, you can do it. We also have the social interaction, so we can place 2 fish per well, and to give the distance between both. So you set up a range, a distance between the 2 individuals, let's say 10 centimeters, and as soon as the fish will enter, they will be close to each other under those 10 centimeters. Distance will be counted as a contact. So we will give you the number of contact, the time spent in that proximity area. And once we see only one gravity point within that well, as we have 2 when there are 2 and swimming around. So as soon as we saw just one, we call it a fusion contact. It means that they are that close to each other that we count it as just one. And it gives the fact that they are really close to each other, maybe like kind of tail-nose behavior and so on. So you can quantify such like a fine behavior, depending also of the pathology of the scope that you are working with. You can have the need to analyze how the fish swim, because sometimes they can have this kind of specific habits to turn clockwise or counterclockwise, or to take different pass angles. So we can classify and count all of those. For the angles, we have like a histogram where you have 12 class angles, and we count it for each class angles that you enter from plus 20 degrees to 45, and we count it how many times they take that angle. And now I'm going to present you and to talk about the different. So before it was let's say the software option, and now I'm going to talk about the hardware. So it's different devices that we implement on the Zebrox or spare units in order to bring different information or stimulation to the Zebrafish. So it's a vibration module. So we stick a speaker connected to an amplifier, also connected to our software, to deliver some vibration, some sound stimulation. So the sound has it stick to the microwave plates. will be perceived as kind of vibrations through the router. And then we can analyze how at this acoustic level, how the fish are answering in terms of locomotion to that noise. Then we have different kind of range, depending how far and precise you want to go on that one. For some labs that are working on hearing loss, they just don't want to have like a just sound stimulation and see if they react or not. They want to measure at which level they start to behave, at which dB they start to have an extra activity, or at contrary, any activity or some stress level that can appear. So in that case, we got this specific request, and that's why we, for example, we improved this vibration module with the sonometers and a probe that we put inside the system to always calibrate the sound to make sure that the sound we deliver on the software is the one that we deliver inside the system. And then we also created this isolation chamber where you place the Zebrox in, and with a dedicated and technical foam to reduce the ambient noises down around 40 dB less. It's anti-moisture foam, and you also have a ventilation device inside to keep aerated the chamber. And recently, we developed that optomotor response test for reflex behavior on fish. And it works that way. We have a HD flexible screen that we place underneath the mackerel plates. And then we displace different stripes of pattern. At the moment, we have two protocols that we can automatize. So the first one is those stripes, black and white, that goes up and down. And then we want to analyze how the fish react with that. So we design in-house at Viewpoints dedicated mackerel plates with 10 alleys, 10 corridors, with 1 larvae per rail. And then on the software, when at the beginning I show you how to create the protocol with the different areas, in that case, we drag those areas per corridor. So 10 alleys with at the both extremities 2 individual location. And the red dot that you see is the larvae that are moving through the plates. And in fact, we will calculate how fast the larvae follow the stripes. I mean, how fast they manage to follow the stripes in the normal direction, and how fast they end up to the extremity. So as soon as the larvae enter the extremity area, the software stop the experiment for that corridor. So then in your output file, you will have for this 10 location, okay, the first 5, for example, my control, they managed to follow the stripes in 5 seconds. When the other half one, the mutants that you want to check something, they needed 15 seconds, for example, to reach at least 50 seconds to reach the bottom navigating through the stripes. We can also, the other protocol that we have using that screen is the fact that we can also, through the software directly, split the screen into 2 colors, for example. So the first top half being white and the bottom one being black, or other colors or anything. And then it means that half of the plate will be under the same condition on the top and the bottom as well. So we can analyze the place preference depending of colors and so on. And we try to also think of different protocols to go deeper on that thing. So we realize that when the larvae are following the stripes, they really swim toward the stripes and really like linearly. So they go straight forward. When they reverse the stripes, they have more random navigation, swimming behavior, going right to left and so on. So it's what we could see. And as you see, we can customize also some plates. So if you have any ideas, for example, we think of putting some inserts on both edges of the plates to put some conspecific in each of those and try to analyze the time spent per the larvae that are fully moving inside the time spent when there is only one conspecific at the top, and at the bottom where there is an enclosure with maybe 3 or 4 larvae. What are the preference in terms of that? So it's something that we can play with, or some experiment we can think about. And here is a video of how it looks like. So you can define the width of the stripes, the speed, and so on. We also have some temperature control units if you need to control the temperature, so which can be important when you really get rid of all external factors and bias. And then also from different requests, we implement it as a stimulation through our software, meaning that when you design your protocol, you can also define some, for example, not light stimulation, but thermal change stimulation to know what are the behavioral response depending on different temperature. For example, out of a 3 hours experiment, the first hour will be at 22, the second hour at 24 degrees, and the third one at 28. And your output file will be split into those 3 different thermal change to see exactly what was the pattern in terms of locomotion and behavior on the thermal change. So you can heat or cool down, so you can think of different protocol. For the optogenetic also, we place top lights with different wavelengths, so up to 4. So you have a few examples of the optogenetic wavelengths that we can place. So the most used, I would say, is the blue one to activate or inhibit dedicated neurons to activate some behavior and so on. So it's something that can be easily done with the system. And for adult fish, as I showed you at the beginning, we have this bigger chamber that works exactly as the Zebrox. So it's kind of like a big fridge, like 1 meter, 1.20 meter height, with white light stimulation. So the same, to have a control environment with infrared lighting system to be able to work in the dark condition when you close the door, with white light inside to either trigger and see some startle reflex based on the light, or to respect the circadian cycle with some day and night protocol, with a top view camera inside. So you can place up to 10 individual tanks on the system and follow all of them, or you can place one big tank for the shooting, for example, with 10 or 15 of adults inside. And then you have the heat map generated. You can see on the left screenshots the trajectometry, so to see as the heat map where he spent the most of his time, but also with the speed color code, you know exactly also what was his activity level depending of throughout the experiments. So red being the high activity, so when he is in acceleration, white when he's more moving around, and green the average one. And all the stimulation, I mean, most of all the stimulation presented can be also implemented in that ZebraCube. So the temperature, we can have kind of bain-marie, a water bath, where you place the tank in and we keep the control, the temperature, the white light system, the vibration. And recently we discussed with Planktovi to synchronize the fish gun apparatus with our ZebraCube system, either for larvae or adult monitoring, to be able to synchronize the locomotion assessment with automatized food delivery, meaning that we can imagine a big tank with 2 different area or compartment within, and see or deliver food when the fish is on one or the other side of the tank. Or it can be just to, because I attended some talk recently on the Zebrafish Congress in Italy, where a scientist talked about the fact that controlling the feeding behavior can have an important role on the reproduction, on the behavior, and that could be like a really interesting point and things to monitor and to control. So that's why we thought about that, and we checked and discussed with Pantobi how we could do it, so we can synchronize both of the system to automatize that. So as soon as the fish will be in area, we will deliver some food, or just along a 2 hours experiment to deliver some food. Take every, let's say, 10 minutes out of an experiment of 1 hour in order to have like a control environment. And as they can also deliver like living food, such as rotifera, Artemia, Daphnia, also dry food, we can also combine toward which feeding the fish are mostly interesting to take, and so on. I mean, we can think. So that's why I take the opportunity just to discuss about that, if you think it can make sense, if it's pertinent, and if it's in interesting things to develop. We can also create different mazes, T-maze or light and dark box on request, meaning that if you have some particular maze that you want with particular dimension and so on, feel free to ask. We can custom those the way we want. For example, for the T-maze, we have some transparent one where we can stick pictures of, for example, one specific hair, and on the left arm a shoal of zebra, and the zebra being tested will start here, and we will analyze toward which corridor it's going to. And based on the literature, we know that the reward can be the food, but can be also just this kind of image of the conspecific or shoal, and the fish will accept that as a reward. And even in two dimension, even not using 3D, the C can distinguish those conspecific and so on. So it's something that can be done also through audio test. And we don't only propose a locomotion tracking system, but also a cardiovascular one to analyze the heartbeats and the blood flow in an automated way on larvae zebrafish. You place them on agarose or methyl cellulose, and then with a trinocular microscope with our high-speed camera placed in it, we can analyze the blood flow, micromillimeters per second, and get also the blood flow. We also are validating a new outputs more along cardiac performance to analyze the beat defects and the ejection fraction. So what is the volume that reject the ventricle and the atrium for each beat, and if there is some arrhythmia problems. So we are validating it. And for that, we need the Hertz videos with GFP, green fluorescence protein, to be able to have a nice contrast and being able to see precisely those things. So for that one, it's what we require in terms of acquisition. And but it can also work with some video that you already have in-house that we can just download it in our software to release the outputs. For example, we validated our software with videos that have been generated from the VAST system from Union Biometrica, but it can be your own system with your camera, and in that case, we will always ask you to send us samples of the videos to let you know if it can work out fine, if the contrast is enough, and so on. We also have some system for the visual acuity and for the oculomotor reflex and muscle reflex, the optokinetic response test. So that test is based on placing up to 5 larvae zebrafish in a star shape. So each of those facing this HD screen that are turning around with those stripes, the black and white stripes, and then we follow and we analyze how the fish follow those stripes with both right and left eyes when they follow it. It's the way the same that when you are driving and you are just fixing a point at the horizon. I'm not sure about that, but a tree or a car or house or mountains, and then you just fix it. And as soon as you get as soon as you cannot follow it, you just get back to your initial position, and so on. So it's what we analyze by calculating the angles from the fish head and both center point of both eyes. So you see that we have two velocity phase. So the slow one when you follow the things, and the speed one when you just retrieve and get back to the initial position. So we can quantify all these movements. So you see how the system looks like. So that was our previous system based on a video projector inside that display those stripes. And now we have this HD flexible screen that I present you on the software for the optomotor response that we use. And therefore the system is way smaller and so on, but we don't have yet the new illustration pictures. But it works the same. So you can calibrate, you can define the speed, the rotation of the drill, the thickness of each stripes. You can also play with the colors and so on. And to conclude, is not based on Zebrafish at the moment, but who knows, maybe we will be able to change it. But the ToxMetLab, it's for long-term monitoring at the moment on aquatic macroinvertebrates, up to 48 simultaneously. So you see that you have 3 different panels. Each of them contains 14 individual cells where you have your organism placed inside. So all of them are within the same water flow condition. The entry tubing is at the bottom and the exhaust at the top. And those tubings, the entry tubings is based, pump some water in a bucket close by. And then all this water continuously goes through the panel. So the species, the organism cannot go from one cell to the other because there is some filters, but the water goes into it. So you can do it on those 3 independent panels. You have 3 cameras facing each of those. And you can work in different scenarios. For example, you can have one single concentration for the entire panels, but per panels you have different organisms or species like that, or different organism stages to know, okay, how this concentration affects the larvae zebrafish at, let's say, 4 dpf or at 6 dpf and so on, to see if there is a difference in terms of behavioral response when the organisms are in contact with such a concentration, pollutant, or whatever. So at the moment, it's not for zebrafish, but as we don't know how it can go. I just wanted to talk about that, and it's mostly to test some water quality or how it impacts their behavior with the white light inside, to always have a control environment and to get rid of all external factors. And so that's all for me. So I would like to thank you for your attention, and to everyone to take your time to participate to the webinar, and Cyril and Planktovi have initiated that webinar session. So if you have any questions, so either you can ask at the end or write me an email, or going through Planktovi will be in touch. And thank you, and have a nice afternoon.
Thank you. Thank you very much, Antoine. We have some many questions. First of all, is it possible to design some other patterns with the flexible screen?
Yeah, the screen. We can design most of the things we want, some different patterns and so on. At this moment, as we just launching it, we in an automated way, we have kind of just this place preference test with 2 different colors or those stripes. But then, depending of how your protocol will look like, then the idea is to be able to do whatever you want. So you can let me know, you can write me what would be the idea you have. But yeah, it's a screen, so basically we can we can define what we want on both sides, or yeah, that the idea, in fact, to use a screen. That way it's really easy to implement in kind of patterns, shape, and to automate it. But then we have to take care also how the fish will perceive it from the plates. That's why we needed to design these 10 corridors to have them in the same condition and so on, because then to calculate and generate the data outputs, we need to take care of, I mean, all the different parameters. But yeah, the idea is to be able to do whatever you want. Is there a possibility to test the Zebrox? Yeah, you can test the Zebrox. I mean, most of the time we discuss we can plan a demo, a call. I can show you how it looks like, and then if you need to test it out, we can discuss it. Yeah.
Thank you very much, Antoine. Thank you for your call, for your presentation of the Zebrafish, so the Zebrabox, and all the other devices from Viewpoint. Thank you everyone for your time and for your time for this webinar today. I hope you have a pleasure to listen us, and we will send you a certificate of participation in the next few days, and some questions to have your feedback about this type of webinar. If you like it, just say us. If you have any questions, don't hesitate to contact us. In our next message, we will send you some our contact and the contact of Antoine if you have some more questions about the Zebrabox or about the spin touch, the fish gun, or other products we can provide you. Thank you for your attention. Have a nice end of afternoon, and see you later for a new webinar. Thank you very much for everyone. Bye-bye.
Thank you, Cyril. Thank you, everyone. Bye-bye.